esteemed eminent speaker, Dr. Indira Kaur. Currently, she is working as professor at University Institute of Sciences, Punjab University, Chandigarh. She has more than 30 years of teaching and research experience. She is independent director of Nectar Life Sciences. She is secretary of Indian Pharmaceutical Association, Punjab. Presently, she is working as project coordinator, UGC, NRC, program UIPS. She is working as president of Punjab University Pharmaceutical Sciences. She is director of VSIC, SAIF, and RSIC. She was Dean Faculty of Pharmaceutical Sciences during 2017 and 18. She is identified for her passionate dedication in the field of research. She is recognized as 2% most cited scientist across the globe as per Stanford University, USA. Her research work extensively on novel drug delivery system, nanotechnology, dermal and ocular drug delivery system to overcome the delivery challenges of molecules like antifungal, antitubercular, etc. Emphasis of her work lies on industrial and clinical translation is reflected through licensing of 4 of her technologies to pharmaceutical, biotechnology, and dermatoceuticals industries. She has guided more than 31 PhD scholars, 58 postgraduate, and 2 postdoctoral fellows. She has served as a consultant and coordinated with more than 10 pharmaceuticals companies. She has delivered more than 75 talks. She has published around 125 high-impact publications. She has written 8 books, 25 book chapters to her credit. She has received US Fulbright Fellowship. She has received project funding of around 100 million rupees from Government of India and industry. She has filed 33 patents, out of which 13 are granted. She has honored with many prestigious awards. Some of them are I listed here. She has awarded as National Intellectual Property Awards for being top Indian individual for patent filing, grant and commercialization, and the WIPO for inventors. She was awarded with Women Scientist Award, Technology Exposition Award, Researcher of Year Award. Recently, she has received funding for project at SCES'S facility, Rutherford Laboratories, UK. With this huge introduction, I welcome you, ma'am. We are fortunate we have a great researcher and scientist with us. Welcome, ma'am. Welcome to the session.
Thank you, Dr. Jyotsna, for a very kind and I think a long introduction we need to. Thank you so much, and I'm really privileged to be here. Thanks to the organizers, my dear, Madhur and Dr. Joshi for having me here. And I would try and take you along with me on some of the lessons I have learned across my professional journey. So I'll just share my screen. Okay, can you see it?
Yes, ma'am.
Okay, so this is what my university looks like. I am from Punjab University, Chandigarh. So this has been a pre-independence university set up at Lahore in 1882 and later on moved to its present destination at Chandigarh. These are 2 hallmark buildings of the university. This is the student center and this is the Gandhi Bhavan, and several of these buildings were initially architected by Le Corbusier. So this is what my department in the university looks like. I belong to University Institute of Pharmaceutical Sciences, and we have a proud heritage of being an institute which was in a sense initiated by Professor Harkishan Singh. He was the chairperson of the institute for a very long time. I think people from the pharmaceutical and medicinal chemistry or drug discovery background would definitely have come across his name. He's the only Padam Shri in India from the area of pharmaceutical sciences, established himself not only as a researcher, but also as a historian. And again, this department is also a pre-independence department, which was set up in 1944. And though belonging to a state university and a very small department in a state university, we again very proudly own NIRF rank 2 and 3 consistently since the time MHRD started ranking the pharmaceutical sciences institutes. Similarly, we are still even very proud of the faculty, which is even currently working, and 12 of us, along with 2 research scholars, are listed amongst the top 2% most cited scientists in the area of pharmacy and pharmacology. And again, though a humble department, we are credited with 1 drug discovered, 8 products in the market, 11 technologies transferred to the industry, and 3 series of new molecules being discovered. So I start my talk with this quote, and I always feel that science is basic to all the phenomenon of life, I would say. It is fun if you find passion in it. It serves to, satisfy our curiosity, process of investigating, posing questions, coming up with methods, answers, and delving into it further. So each idea, each research work, each curiosity blossoms and branches into newer areas, and the quest goes on. So, and today's topic, which was assigned to me by the organizers, were to talk about the translational research. And so what is translational research? So the entire idea is to produce. So what is the difference, I would say, from basic research? Basic research is to determine underlying nature of things, trying to find answers to some questions. But when you apply that information to do something for the general improvement of human health or mankind, that is what translational research is, where you move the discoveries quickly and efficiently into practice. And similar is something when you define a patent, and it is said that it should have 3 prime things in it. One is novelty, one is inventive step, and the third is industrial applicability. So it is something again to do with translational research, that it should be translatable to the industry and to the clinics at a faster pace for the mankind. So what are the key points for being innovative or doing translational research? Is you have to be creative. Try to introduce better and less expensive products, technologies, or services, and the time to move to the market should be small. Again, I have this quote that the science of today would be the technology for tomorrow. It would be a new product or a service or a technology for the generations to come. So from pharmaceutical sciences, where do we fit into translation? So we've been talking of science, technology, engineering, and mathematics. And if you add art into it, the art of, preparation with pharmaceutical art, research with creativity, and that is what STEAM is. And I'm sure that is where we, as pharmaceutical scientists, find ourselves. So we are multidimensional, we are a multidisciplinary branch. So till several years when we were young, when we were students, I'm sure the young teachers and the students who are amongst the audience, we've always been telling ourselves that we are jack of all trades, because we were taught chemistry, we were taught engineering, drawing, we were taught mathematics, we were taught physical pharmacy, which even included some concepts of physics beyond pharmaceutical technology. So not only pharmacology, but physiology, anatomy, and so forth. So we have been thinking that we were jack of all trades, but no. Let me tell you, today, when we are talking of science, of industrial applicability, of modern science, then people are doing away with all the discriminations between science. अगर हम शुरू की बात करें, the historical background जब हम पढ़ते हैं, so we talk of people like Louis Pasteur. So who was he? He was basically a chemist, ventured into microbiology. Madame Marie Curie, she was given 2 Nobel Prizes, one in physics and another in chemistry. And she's still now the only Nobel laureate to receive. There are people who receive 2 Nobel Prizes, but to receive 2 Nobel Prizes in 2 different subjects, that was what Marie Curie was, what makes her different. So I think we have to do away with the boundaries between science, and that is where we find ourselves in a better position than people from other sciences. And then most of our science relates to life like incidents. I would say we have been learning from our folklore or the observational data which was there, things like that. So any science is formed from keen observation and a proper data analysis. And several of the pharmaceutical sciences aspects are attributed to those observations from life. For example, I always give this example to my students. मैं जब उनको microbiology पढ़ाती हूँ, I talk about the osmotic pressure and its significance and how it has to be specific. And then I always talk of the pickles, and I always say, how are they preserved? The dadis and nanis have been preparing achar at our homes, and they would last us several years. So what were the reasons? So there were 2 things. One was they would add salt, which would increase the osmotic pressure. Another thing was they would load it with oil so that anaerobic conditions would exist inside the pickle. So that is what I'm trying to tell you, that we relate very well with different facets of life. And we have a role assigned to us by the society. For pharmacists, they are a bridging gap between the physician and the patient. And we have been assigned to influence lives, to talk to patient, interact with patient, look for their comfort. In whatever arena of pharmaceutical sciences we are always concerned with the way we can make the human suffering lesser, improve the quality of life, and things like that. So my vision. So this is a type of a motto or a logo I designed for my labs a few years ago, and innovation to reality. So whenever we talk of innovation, there has to be some research going into it. And how does that? So this slide is very dear to my heart. I'm sure in some previous talks at ICP or anywhere else, I've always included this slide. So you have to start. So research can be compared to an hourglass. You start from a broad base, which is your literature pool. You look into all the prior art and the public domain data which exists. And now, when I talk of innovation, I would always suggest and advise that we need to include patent search into our literature search, to look at what already exists in the prior art, in the public domain. So based on that, then we need come to the nozzle of the hourglass, the thin part. And that is when we are able to identify the gaps in the existing knowledge, the lacunae, what we can address, the questions we are asking and the answers we are looking for. And that gives us our hypothesis, our rationale to do our work, our research. And that is when we propose our hypothesis. And once we do that, we need to, but before that, rather than making ambitious agendas, aims, or objectives, we must look back at the resources we have in our institute, the type of equipment, the type of competency we have, the type of manpower we have, the type of technical support we have, the type of infrastructure we have. But here we can also, we should not be, at the same time, I would not say that we should be dejected or disappointed at the resources we have. Then we should look at the neighboring institutes, the collaborators who can support us in some way. But at the same time, we must have some basic infrastructure with us, and accordingly, we should propose our aims and objectives. And the other day, I was also discussing with Dr. Vanna Patravale, and it doesn't have to be very complex. It does not have to be very complicated to be novel and innovative. It can be kept simple. And let me tell you, it is always like the answers are always simple. We tend to make the things complicated. So if we are looking at the answers, we must look at the simpler versions, because nature जो है वो बहुत systematic है, organized है, and all the answers do exist in nature. So I would come to this part of my thought process later as well. So what I wanted to say is we can keep the things simple. We can look at the resources we have. Am I audible?
Yes, ma'am.
Okay.
Yes.
So based on that, then we can design our methodology. So this is again something which I always want to tell my audience and my colleagues and my students is कि methods or methodology में भी अंतर है. So methods is what we are actually doing, the experimental design, but methodology is why we are doing it. What are we trying to achieve? So methodology is something which matches with our aims and the type of results we are expecting. So that is what methodology is all about. So based on that, we would get some results. And as I said, you have to be a keen observer. So many a times, by your keen observation, you can identify things which you were actually not looking at, but you found them, which could be the serendipity. And we all know that lots of discoveries and inventions are owed to serendipity. And serendipity was where someone did observe certain, things which were not expected. So then based on those observations or data analysis, either our hypothesis is proved or disproved. And this is again something which we as teachers must be aware of. So rather than, pressurizing our students, or rather than demotivating them by saying nahi tumhare to sab results galat aa gaye, we need to sit with them, we need to reassure them, and we need to then discuss with them what results they got, how they are different, and then we need to go back to our hypothesis to confirm whether what we proposed was correct. Then we need to check our methodology, whether the type of experiments we performed were correctly designed to address this hypothesis. And before that, we need to also confirm the reproducibility of our results. So one-time results, one experiment should not make us panic, and we should try and repeat the experiments at least 3 to 4 times before concluding that the results are not matching with the hypothesis we proposed. So once we know that the results are correct, then we need to look at the answers. We need to find the answers why we are not getting those results. So before that, there could be a situation where we feel that we need to redesign our experiments. Then we can do that, get the results again, see whether they justify the hypothesis or not, and then start looking for the answers. And again, I would reiterate, the answers are pre-existing. in the nature, in the public domain, in the literature, we just probably were not looking at the right place, or were not looking at it at that time when we went through that literature. That is my personal experience as well. And anyone could, I'm sure all the researchers here could vouch for it. And at this point, I would say it is research. So it is something which is already existing and we are trying to find it, or we are trying to find answers for something which is pre-existing. So if it is pre-existent, there has to be an answer. But again, there could be a situation where we are not finding the answers. Then in that case, again, I would suggest that those negative results must be recorded, must be published, and must be brought into the public. Because, if my thought process is working in a certain way, I'm sure people within our country and across the globe are also working on a similar hypothesis. So my reporting of the negative results would save time, money, and manpower of another lab, another researcher, another institute, another, master's student or a PhD student. So that now they would know that this way of experiments is not answering the question. We need to redesign the hypothesis or redefine our goals. So that is what the negative results would do. So my lessons. So the first thing I need to tell everyone is that we should not be discouraged at any moment of time. And we should never stop our quest of whys, hows, and wherefores. There could be situations when we would not get the answers. There would be situations when we would have failed experiments. There would be situations when we applied for grants and we did not get them. So there could be n number of situations where we would want to quit. But let me tell you, we need to continue, because ये नहीं पता हमें कि कब हमारा tipping point आ जाएगा. I'm not sure how many of us have read the book Tipping Point, where they have reported several studies, and there have been, so tipping point, what is tipping point? So it is, we all know of the modal graph. We have been talking of the normal distribution and things like that. So there would be a situation when we are going like this, we are putting in efforts, and then there would be a plateau and then a downslide. And that downslide here is reaping the benefits of your work. And you have to just do one little bit. जो हमारी balance होता है, तकड़ी है, तुला है, उसमें you, all of us know, just last one speck, one grain, and the balance goes down. So we very many times just fall short of that last speck, and we lose our confidence or our perseverance, our consistency at that point. So please keep trying till you reach your tipping point, which could be very close, which could be a little far, but it would definitely come. So have confidence in yourself. Each one of us is gifted, and we need to, try and attain that gift. So at this point, I've already told simpler the innovation. So we need to keep it simple. We don't need to complicate the things, because simpler it is, more potential it would have for being translated. And we have to keep in mind, coming from pharmaceutical sciences, where a large part of it goes into technology, may it be from any area, whether it is drug discovery, whether it is drug delivery, whether it is preclinical studies, whether it is any other branch, PA, QA, whatever. So we all know that we have to partner with the industry. And for partnering with the industry, we all know that the industry would have its own limitations. May they be regulatory limitations, may they be limitations of the infrastructure, may they be limitations of the machinery they have, and things like that. So be sure that you try to pre-think, you try to uh, think, you try to develop a vision beyond what you are trying to do in your research or science. I know it is very gratifying to do something new, to have experiments and get the results. But coming from an applied branch of sciences, we must try and foresee the limitations and the way our research can benefit the society and reach the market. For that, we need to look at the technology or our methods which are scalable and industrially viable. Then this logo, as scientists or researchers, we always have this fond desire to publish. So, but we should try and go beyond that compulsion to publish. We should look at our results, at our data, and see if there is any novelty or an innovative or inventive step in it. And we should then think of first filing a patent and then trying to publish it. And I think patent filing, maybe there is somewhere a mental block with us where we think that we cannot file a patent, or we think kaise karenge. So we just need to take it one step at a time. And there are so many, we all know of provisional patent filing. I think now there are so many courses going on patent filing. There are so many soft data available on the net, and the Indian Patent Office has also become very compliant and very, I would say, user friendly. There are their site is very user friendly. So we should try and file a patent application. We can always connect with a patent attorney. There are so many agencies, including the National Research Development Corporation, which is DSIR sister, or I would say a subsidiary of DSIR, who help you to file patents, at least in India. But for all of this, we need to step out of our comfort zone. And the comfort zone is to do the work and publish it. So we need to get out of that. Then try to look at some role models within your profession, within your institute, or across the globe, and then try and develop those skills or try to gain the qualities they had. So rather than thinking of what to do, we should move forward on how to do it. We should say, I have to do this, and then take baby steps on how to doing it, and expand your partnerships, may it be within the institute, within the institutes, the neighboring institutes, within institutes in India, across the globe. There is no limit to collaborations. And I again re-quote something which I always quote is, हम लोग बड़े कई बारी tight-fisted रहते हैं, या we have our own, I would say, we have our own apprehensions about collaboration. वो मेरा idea चुरा लेगा, वो उतना काम नहीं करेगा, उसने 50% काम नहीं किया मेरे साथ. लेकिन मेरे को ऐसा लगता है कि अगर मैंने 99% काम भी कर लिया, मेरा वो 99% 100 नहीं बनता जब तक मुझे वो 1% किसी की कंट्रीब्यूशन नहीं मिलती. और अगर मुझे 1% कंट्रीब्यूशन के लिए भी किसी के साथ कोलैबोरेट करना पड़े तो क्या हर्जा है. एंड as I said, science is becoming multi-dimensional, and there are a lot of things happening now, everywhere. So सब कुछ करना पॉसिबल नहीं है, और मल्टी डायमेंशनल होने की वजह से मेरी साइंस ट्रांसलेटेबल नहीं है जब तक उसमें डिफरेंट वो स्पाइसेस नहीं पड़ते, वो डिफरेंट कंपोनेंट्स नहीं आते उसमें. सो उनके लिए मुझे हमेशा ही किसी ना किसी के साथ कोलैबोरेट करना पड़ेगा. एंड फॉर माय women scientists out there, they may be girl students, and so there are no limitations to science. And as Kiran Mazumdar Shaw, she said, I have never let gender get in my way. So science जो है उसमें कोई boundaries नहीं है, कोई bias नहीं है, कोई dogma नहीं है. तो वो इसलिए मुझे ये कहना है कि साइंस जो है जेंडर के साथ लिमिट नहीं हो सकती, रिसोर्सेज के साथ लिमिट नहीं हो सकती. रिसोर्सेज की वजह से मैं कुछ टेक्निक्स नहीं कर पाऊंगी, किसी तरह का डेटा नहीं जनरेट कर सकती, लेकिन मेरा माइंड, मेरे आइडियाज, मेरी नोवेलिटीज, मेरे सोचने की क्षमता, मेरा इंटेलेक्चुअल जो प्रॉपर्टी है मेरे माइंड की, जो मेरा इंटेलेक्ट है, वो किसी से कम नहीं है. सो इसलिए मुझे कुछ भी लिमिट नहीं कर सकता. So science gives us emotions when we really get to know it, and that's when it starts giving and growing within us. So these are a few other things I would like to talk. So do your homework, read, and read. There is no alternative to reading, and that is what formed the broad base, a broad, a start of any research, any novelty, any innovation. So you need to read the data. And I had reached at the nozzle, and I did not talk about the broad base. And that broad base is the branching of your research, your results, opening of newer arenas. You start from one thing. You do a part of your work with one master's or one PhD student, and then it starts diversifying into so many other areas which you want to do. So there could be one new technology you've developed, that for that you need to do some preclinical studies, you need to do some toxicity studies, you need to work on its dossier for regulatory filing. So the same single idea can branch into several things. Or while you were evaluating a molecule or doing some studies, you realized it could be used for another application. That is what repurposing is. That is where drug discovery steps in. So there is no such thing as good luck. We always say that science is more to do with luck, the innovation or discoveries. It has always been said that it is more to do with luck. Sometimes the lady luck, it shines on you, but no luck comes without hard work, let me tell you. So hard work is always a prerequisite for doing good research, for producing innovations, for doing translational work. And keep trying. That I've already told you, and I always, so this story is also very dear. I'm sure those of who have listened to me earlier would know of this story, my telling this story about King Robert Bruce, who once, lost a battle. He ran from the battlefield and was, hiding in a cave, and he found a spider which fell off its web, and it was trying to climb through the web thread, and it would climb a few steps, and then it would fall back and again climb, and eventually it succeeded and reached its web. And from there, this king, he got his motivation, his idea that I need to try again. So he reconsolidated his army, and he again fought the battle against his enemy, and then he won the battle. So keep trying till you have your win innings to your credit. And there is no alternative to passion. And either you love your work, you have a passion for your work, or you already have that passion, or if you are not, you did not select your work, your workplace, or your profession, then try and develop that love for it. Then make your peace. Each one of us have our own calling. So you have to see what your calling is. You could be happy doing your teaching. It's fine. There could be a colleague who is just happy doing her teaching, but is not into research, is not writing for her projects. She does not have any funding. In the Indian system, there is not much difference between a worker and a non-worker. Once you are into a job, you are getting the same salary, you are getting similar promotions. But try and make your peace that what I am doing for my gratification. So there would be times when you are discontented or disillusioned by the system. But at that time, then I need to, remind myself that this is what I want to do. And I am doing it for myself and not for anyone. And I have competition with myself and no one. You can do anything if you think of it. So keep trying, keep doing. Then another thing which is very, very important is sincerity, integrity. And one more thing I would add here is data, good laboratory data. So good laboratory practices, maintaining data. So this we have to teach to our students as well as learn it ourselves. Because whenever you are thinking of translating your work, सो ट्रांसलेटिंग में सबसे पहले रिप्रोड्यूसिबिलिटी बहुत इंपॉर्टेंट हो जाएगी, फिर उसकी स्केलेबिलिटी आएगी. सो मेनी अ टाइम्स वी डू एक्सपेरिमेंट्स in our lab, our students report certain thing, but if we are not confident of it, we must go back, check, and we must teach our students as well to be sincere to science, to report only those things which are correct. And that is where that thing came into picture कि हमने कभी force नहीं करना अपने student को कि तुम्हारा result ऐसा क्यों नहीं आया, because that would lead to sincerity. If I do not force my student and I sit with him or her to explain the data, to reassure him or her, and if I just scold the student, she would be forced to forge the data or bring a data which I am more comfortable with, which I think and expect it to be there. लेकिन वो फिर डेटा जो है मेरा ट्रांसलेटेबल नहीं हो सकता, बिकॉज इंडस्ट्री में मुझे प्रूव करना पड़ेगा जाके कि जो मैं कह रही हूँ वो है, वो होगा, वो स्केल अप भी होगा. सो in spite of being very confident about your data, there are so many hiccups on translating the data. I give you one example. So we did some nanoparticle technology thing, and then we licensed our technology to an industry. Now when we tried to reproduce it at the industry, it would not work. And let me tell you the type of sleepless nights I had, the type of heartburn I had. The student who had worked on it had already left several years ago. Another student was working on it. She was equally disturbed because we were unable to reproduce the things. And then we realized कि मैं जो काम कर रही थी, so this was working with tretinoin, all trans retinoic acid. So मैं जो retinoic acid use कर रही थी वो सिग्मा का था, वो 99.9% प्योर था. और जो कंपनी मंगा रही थी वो एक वेंडर से प्रोक्योर कर रही थी. एंड फॉर मॉलिक्यूल्स लाइक एट्रा विच आर हाईली प्रोन टू फोटोडिग्रेडेशन, ऑक्सीडेशन, हाइड्रोलिसिस, नाउ दिस बिकेम अ वेरी इंपॉर्टेंट स्टेप. Similarly, the type of surfactants I was using, I was procuring from one type of vendor. It had standard components, but it's peroxides or other impurities होती हैं उन surfactants में, which could, in itself trigger the degradation of my API. So those types of hiccups and problems are already existing in trying to translate your technology. But you cannot be faced with a situation where you are not sure whether your data is reproducible or not, whether your data is reliable or not, whether the integrity of the data has been maintained or not. So that is why it is very important. कि वो कहते हैं ना बिजनेस में बेईमानी नहीं हो सकती. सो सेम इज ट्रू फॉर लाइसेंसिंग ऑफ टेक्नोलॉजीज एंड ट्रांसलेशन ऑफ टेक्नोलॉजी. देन बियॉन्ड दैट, so clarity of thought and speech, I feel it connects with the homework I do. So the more I read, the more I study, the more answers I get. When I try to analyze my data, I try to look at, I read literature. So I always get the clarity of thought and speech because I'm confident I have done my homework. then I am a strong believer. आप उसको nature कह लीजिए, आप उसको supernatural part कह लीजिए, आप उसको God कह लीजिए, आप उसको ether कह लीजिए. मुझे लगता है बड़ी बारी, each of us gets our eureka moments. जब हमें suddenly ether से कोई message आता है या कुछ strike कर जाता है. और answers जो हैं, जैसे मैंने पहले भी बोला, they always exist, So, and here I bring out this, quote where there have been across the years in the history also, there have been times when the scientists, they have been, scrutinized and assassinated by religion, by church, or by anyone. But at the same time, scientists have always been claiming that they are the most religious people. So science and spirituality, they are not exclusive. The spirituality is inclusive in science, I would say, because we tend to respect nature. We know that what is happening, we do accept what is happening and then try to look for the answers for it. So that is where the connect is, I would say. So what did I achieve? So there was a time when I did not have much infrastructure in my institute or in my lab. I was a young faculty. I did not have access to the equipments which were there. And then there are always boundaries between sections, between departments, between within the department and beyond the departments. So that was a time when I took it as a challenge. I started writing my own grants. And again, I tell you here, we do not have to be discouraged. We write one grant, we say, wo to mila hi nahi. Fir badi bari, I know it happens कि जो comments आते हैं वो बड़े vague होते हैं, कि वो critical comments नहीं होते कि हमें पता लगे कि क्यों हमारी जो grant है, जो हमें funding नहीं मिली और क्यों हमारा proposal reject हो गया. But इससे आगे ही जीत है. So keep trying, keep writing new proposals. You could connect with some mentors, with your senior people within your institute, across the institutes. Talk to them, write, rewrite, think of newer ideas, propose them, and you would be successful. Then many a times, we have this कि वो तो जान पहचान से मिलती है grant, और किसी का गॉडफादर है उसको मिल गई. बट लेट मी टेल यू, मेरा कोई गॉडफादर नहीं था, कोई मेंटोर नहीं था. तो मैंने क्या अपने मन में सोचा कि कुछ तो जेन्युइन मिल रहे हैं, 25%, 30%, 50% या 10%. सो आई एम जस्ट कंपीटिंग फॉर दैट पार्ट वेयर I would get a funding on my merit, on the merit of my proposal. It has got nothing to do with my connections, the people I know. It doesn't have to happen that way. And I'm sure you would find so many examples across the country where people would say that they did not know anybody in the evaluation team, in the expert team, and they still got the funding. So, and the other thing is, आज की डेट में भी मैं एक साल में 10 से 12 grants लिखती हूँ, जिसमें से 1 या 2 मिलती हैं. So, and we all know the number of grants which the funding agencies are getting. Those of me who are senior like me, they would know कि funding agency के पास भी अपनी limitation होती है, and 5 से 10% का ही success rate होता है उनके पास, depending upon the grants they have. So 90% ने तो reject ही होना है ना. So we have to just try and come into that 10%, and then out of that 10% also maybe into if we believe, if we think there is some other factors, then further from that 10% I have to compete in that segment. So I could what I wanted to show here was not to boast it, but to motivate all of you that there is scope. You can get grants, you can get industrial projects, and you can consolidate your own lab, your own infrastructure, and do good work. So again, this is the type of awards and accolades I achieved. So what do I suggest? So mutual capacity building would be very important, collaborations and joint undertakings. Do I have 5 more minutes, Madhur? I would compile in that. I think I'm
Yes.
running. Okay.
Yes, ma'am.
Okay, so you have to look about the industrial, amenability of your work, and try and look for international opportunities as well. So here I thought one area where some of you, if you want to look at an area where one can work into. So I thought we have a rich folklore, and the thing is to find a scientific evidence to the observational data which exists in our folklore. And that is where phytopharmaceuticals come into being. I have been working in nanophytopharmaceuticals. So what is a phytopharmaceutical? Is a phytochemical where how do you categorize it into a phytopharmaceutical? You need to have at least 4 fingerprinting tests for it, at least 4 markers for it, then only does it become a phytopharmaceutical. So here we have a person who's asking that how do I make drugs out of the phytochemicals? So we all know that phytochemicals are some things which are made by the plants for their own protection from pests, from animals, and from human consumption as well. And they are effective at small amounts sometimes, but sometimes you need bigger amounts, and that is where the limitation comes of clinical translation of the dose. They have antioxidant, anti-inflammatory, they could mimic hormones, they could alter blood constituents, they have n number of applications. We already have examples of phytochemicals turning into drugs, especially the anti-cancer drugs. We know of Taxols, we know of vincristine, vinblastine story, camptothecin, and podophylletoxin. So I just brought in one example. I have been working with several phytochemicals. I have worked extensively with one molecule, sesamol, which is obtained from sesame oil, tilka tel, we all know that. And it is not naturally present in sesame oil, but sesame oil is rich in several antioxidants. One of them is sesaminol, and sesamol is a derivative of sesaminol, and it is obtained by roasting of these sesame seeds. Why I picked it up? So what I want to consolidate here is that you have to develop your rationale, your hypothesis. So we once screened several, natural and synthetic antioxidants, and sesamol I found was a very good performer in a battery of antioxidants, where it showed 1.5 times the activity of vitamin C, which we all know is a beautiful antioxidant. Then beyond that, in the a tissue system, it was found to, lower lipid peroxidation, manage hydroxyl radicals, and also superoxide anion radicals. Then it was an interesting molecule in the sense that it would just satisfy several of the Lipinski's rule of 5 for the drug molecules. It had low molecular weight. Log P was 1.29, which was again favorable, and the hydrogen bond acceptors and donors were good. So small size and log P would also assign it a suitable permeability. It is known to be a good metabolic regulator, possesses anti-aging and anti-mutagenic activities. So we did establish its anti-mutagenic activity in my lab, and also the anti-aging activity. Then it is known to have several of these activities. The ones highlighted here are the ones we established in our lab also, where we tried to so I always talk of a pharmaceutical couture. मैं कहती हूँ मैं molecules को ऐसे फैशनेबल कपड़े पहना देती हूँ कि वो बायो परफॉर्मेंस उनकी एनहांस हो जाती है, एंड दैट इज I give them a couture. And being a formulation scientist, I usually work on the drug delivery system. So what were the issues with it? It had a low oral bioavailability, though it was not very low, but there was rapid elimination as conjugates. It had low plasma concentration. So we could not get a sustained release. It was eliminated faster. And it was, since it is a phenolic compound, not a polyphenolic, but a phenolic compound. So 4 stomach irritations were reported with it, and 4 stomach cancers, but in humans you do not have a 4 stomach. But in spite of that, being phenolic, you could expect some irritation and from it. So this is what we could achieve. This is part of the publications. We worked extensively with sesamol over several years for different masters and PhD students, and we kept including it into different type of nanoparticles, and we could do some studies on skin cancers, in neurodegeneration, in hepatotoxicity, and this was also in cancer, and we did some work in gastric cancer. Recently we have communicated some work on skin regeneration. So the thrill of being an inventor is to see the creation of the brain unfolding into success. So your idea, your concept coming actually into play is what gives you the thrill, and they make you forget everything. So but at the same time, what is current today would be challenged very soon, and that is where what I talked of at the very start is that the fast change from the lab, from the research to the industry or to the market is important. And in each one of us, there is a genius. In each one of us, there is a hero. In each one of us, there is a scientist who can ask questions and do the work. So the way to get started is to quit talking and begin doing. And that is my final message to all of you. Thank you.
Thank you, ma'am, for your valuable research insights. Now the session is open for questions. All the participants, put your queries in chat box. So we didn't find any questions, ma'am. Where is here? I think you have covered 100%.
I'm not sure whether I heard what was expected of me, but
ma'am, it was excellent session. And
Yes, ma'am.
all the participants, if you can't think about any question right now, please feel free to write to us so that we will forward the questions to ma'am, and we will forward her answers to that. I think, ma'am, what the in depth of work which you have actually carried out and which you have shown, and as you rightly said, we should quit talking and start doing. So I think this is instilled in our minds also right now. And yes, definitely, if there are no questions, then we will participants.
Ma'am,
Yes,
I have a question for Professor Corming, and highly exhilarating session. And for researchers like us, it's really what you can say. I mean, it just gives us a push, and it just gives us the motivation to keep working and to keep on doing our bit. So I had one question, ma'am, like translating your research work to industry. It involves, I guess, a lot of networking with the industry. So how do you make your work visible to the industry? How do you get industry interested in your work? That is my query.
Very apt question, Madhur. So in my previous presentation, I had covered that, but since I had already delivered that talk at your institute, so I did not include it. So you are very right. So that is, important thing. And the factor which actually plays there, as per my experience, I know we have Dr. Vanna Patrable with us. Maybe she can also throw light on it. So what I found was the trust factor. That is very important as far as, translation or partnering with the industry is concerned. So that is where, as you rightly said, networking comes into play. And again, we cannot be, I would say, jaldbazi nahi ho sakti. We cannot be, it takes several years. So let me tell you, the first licensing I did was actually with an alumnus from my institute. He was also our student, but he kept visiting me, coming to my lab. And it took me almost 4 to 5 years before we could come to a situation where he showed an interest in licensing the technology. So it is a very slow process. The fruits are very late to come. But then again, my only message to everyone is, we have to start, first of all, we have to step out of our comfort zone, start taking the first step, the baby step, and keep doing it till you reach your tipping point. और उसमें हर एक, there are no generalizations, हर एक का अपना experience होगा. मेरे केस में क्या था, मेरे केस में सबसे बड़ी limitation मुझे ये लगती है that I am at a place, Chandigarh, where we do not have very many pharmaceuticals. We are not, the strategic positioning is missing for a pharmaceutical institute at my institute. There are no R&D companies in and around Chandigarh. So while sitting in Pune, that limitation will not be there. But you need to go out. You need to, what I would do is I would prepare small, write-ups. Then उस वक्त तो मुझे ये भी नहीं पता लगता था कि मुझे ध्यान रखना है कि मुझे सब कुछ नहीं बताना इंडस्ट्री को. So you have to make a non-controversial, 4 lines, 10 lines about your technology, and then start sending it out to people you think could be interested in it. You have to identify the industries which could be interested in your technologies. And let me tell you, there is a difference between the cup and the lip. बड़ी बारी discussions शुरू होती हैं, बड़ी बारी काफी आगे तक discussions जाती हैं, but then sometimes nothing happens. Suddenly the industry loses interest. So what, I don't know, there could be a number of reasons. One could be that you disclose so much and they realize कि हम खुद कर लेंगे. Other could be they find the market is not ripe for this type of technology. So there could be anything, or they are not convinced by your technology. So there could be anything, I would say. I don't say that हमेशा मेरे में ही खासियत है और उनमें कमी है, लेकिन जो भी है, there are always points when it doesn't happen. But you just need one break, the first break. That is what we have to focus on. अर्जुन की आंख जो है वो, उसकी जो फोकस है वो वही होना चाहिए कि मुझे वो ग्रैब करनी है अपॉर्चुनिटी. I think that's
Thank you so much, ma'am. No, that's what I mean. After what you have spoken, I feel that one sentence, वक्त लगेगा पर हो जाएगा.
हो जाएगा.
It's
Yes.
that kind of
नहीं,
thing what you are
करना
probably
ही है,
insisting.
मधुर करना ही है. हो भी नहीं जाएगा, मुझे करना ही है. So अगर उस तरह की वो लेके चलेंगे ग्रिड ना, तो फिर I'm sure ways would keep coming. God is always helping.
Correct. Yeah, right. Thank you so much, ma'am. Highly motivating.
Yes,
speech.
absolutely.
So if one of you is even motivated, my job is done, then I'm very happy.
Yes. One day, actually, something will come out of us if we can.
Yes, definitely. Each one of us.
Yes. Jyotsna, you can take over the session.
Yes, ma'am. Thank you, ma'am, for your valuable insight. The session was excellent, full of informative, madam.
Thank you.
Thank you, ma'am. Ma'am has told us that how we should be multidimensional, how we should enhance our research process by going beyond the boundaries, by doing the collaborations with others, how we should make our innovation, that the simpler the innovation rather than the complications. Thank you, ma'am. I extend my heartfelt gratitude to spare your valuable time with us, ma'am. Thank you.
All right.
Good morning, Dr. Anuradha.
Good morning. I am afraid I'm facing a technical glitch. Just now, around 10 minutes back, my laptop sort of conked off with the message that my charger is not getting recognized, and error message is continuously coming. It's not opening. So what I've done is I've given my presentation to Madhur Kulkarni, and she can share it. The only issue is I'm going to see it in my small screen of my phone, but I will manage.
No worries, ma'am. Yeah, Jyotsna, please take it forward.
Yes, Dr. Anuradha, I'm absolutely ready with your presentation. So just chill. We will go. We will do a good job. You can just go on telling me when I need to.
You just take my prompt when I do like this. You just, otherwise I'll be saying next. So I'll just raise my hands and change. Yeah.
No problem. Yeah, I'll do that. Only thing is the host, as in Samir or Pooja, I think you'll have to give me the screen sharing rights. That's the only thing. Yeah. So then I'll start sharing the screen.
Yes, Madhur ma'am, shall we start?
Yeah, please, please. Let me just see whether I can share the screen once and then.
Okay.
Yeah, can you all see the screen?
Yes, ma'am.
Yes, ma'am.
Yeah. Okay, great. So then we can go ahead. Yes.
Okay, okay. Thank you, ma'am. We have eminent speaker for today's second session, Dr. Anuradha Majumdar ma'am. She is currently working as associate professor, Bombay College of Pharmacy, Mumbai. She has completed her PhD in pharmacology. She has more than 24 years of teaching and research experience. She is former Dean Faculty of Science and Technology, University of Mumbai. She has guided 10 PhD and 69 postgraduate scholars. She has more than 37 international and 10 national publications to her credits. She has authored 1 book and 8 chapters to her credits. She has filed 5 patents, out of which 4 are granted. She has 3 trademarks to her credit. She has presented more than 64 papers in different international and national conferences through UGC Travel Grant, UK, Taiwan, France, and USA. She has received around 1 crore funding for her 14 projects from different funding agencies like AICT, UGC, DBT, DST, and University of Mumbai. She has received so many awards. Recently, she has received this year Invention All India Gold in Health Sciences and all categories award. With this huge introduction, I welcome you, ma'am, for this session.
Thank you very much, Jyotsna ma'am. Greetings to the leaders of APTI Women's Forum, Professor Vandana Patravale. It is a tall order for me to speak after the lecture of Professor Indu Palkor. Greetings to Dr. Anika Joshi, Principal, my colleague Madhur, I can see Dr. Supriya Shidhe from University of Mumbai, all my dear colleagues. Please bear with me today. I was also a bit perplexed because suddenly the laptop is giving problem, but I'll try to do justice to my talk. So while, as Dr. Indu was telling that, it is a challenge that, for doing pharmacology research, because it is highly resource intensive, you need good funding. And although we collaborate, many times if we can, if you're not able to do in vitro screening for your molecular mechanism or your target, it is good to look into network pharmacology. As you are all aware that systems pharmacology or network pharmacology looks into multiple targets, as we are aware that most of the diseases which mankind is facing, which is threatening, there are limitations to the therapeutic options available to us, are polygenic disorders. There's not one gene, which is a rare case. There are few diseases like, let's say, cystic fibrosis and other glycogen storage diseases, etc., which are monogenic. But most of the disorders which we know, whether it is diabetes, metabolic syndrome, cancer, they are polygenic in nature. Multiple targets can be, looked into, because multiple genes and biochemical pathways are involved in the genesis and progression of the disorder. So my title is Integrating Network Pharmacology as a Vital Tool in Bioinformatics, a Device-Full Approach in Drug Discovery. I'll try to be quick because I have 45 minutes of time allotted to me, but at the same time, try to drive the point that these tools can be easily used by young and all other faculty in pharma, when you are initiating your research. But remember, when you are doing in silico methods or bioinformatics, it's always good, if you want your paper to be published, to supplement it with actually cell line based studies or in vivo studies, so that you can consolidate your case. Next. So what is bioinformatics? Bioinformatics is a subdiscipline of biology and computer science concerned with the acquisition, storage, analysis, and dissemination of biological data, most often DNA and amino acid sequences. Today, we are learning about big data analysis, data analytics in healthcare, data analytics in drug discovery. So today, use of softwares, use of computer-generated algorithms is very important as a tool for research. For example, in COVID, the propensity of the virus for the subsequent mutation can also be deciphered by using bioinformatics tools. The weak links in the genome, which is prone to mutation, these can be deciphered easily by using bioinformatics. Bioinformatics uses computer programs for a variety of applications, including determination of protein function, establishing evolutionary relationships, predicting the three-dimensional shapes of proteins, etc. Next, Madhu. Yeah, so this is again the same thing. So what I would like to concentrate is what we have done. So the application is a computational technology. I would like to tell my dear colleagues from all branches that this is no rocket science, and we all can adopt these bioinformatics tools. Most of these softwares which I have used in my studies are free, so we can use the tool as a prelude for initiating your full legwork. Next. So it is a cusp. So you can see the sets and the subsets. Bioinformatics is a cusp of statistics, data science, computer science, computer biology, and biostatistics. So you can utilize this tool when you are initiating your study to add power to your rationale and your hypothesis. Next. So applications of bioinformatics, as I mentioned earlier, also DNA sequencing, analysis of mutation in cancer, analysis of gene expression, analysis of protein expression, analysis of cellular organization, protein structure, protein function prediction, protein ligand interaction prediction, and protein-protein interaction prediction. Next. One of the most commonly encountered example which we see our colleagues in medicinal chemistry adopting is molecular docking. Molecular docking is one more form of bioinformatics, and in fact, there can be amalgamation of pharmacology research with medicinal chemistry, as you're all aware. Today, it's more true that there is, the research should be borderless and seamless, and between departments, between branches, we should come together and execute projects which have broader outcomes, where you envisage broader outcomes, and that is only possible when pharmaceutics department, analysis department, the chemistry and pharmacology teachers come together within the same institution or across the institutions. And today, if you see, there's also a lot of scope for collaborating with engineering colleges, technology institutes, wherein we can get a lot of inputs from them regarding AI, ML tools, which we can actually generate or curate with them, with respect to pharmaceutical science. Next. So databases in bioinformatics are in plenty. You can see the right panel, Emboss, PDBJ, so many of them. So many databases exist covering various information types, for example, DNA and protein sequences, molecular structures, phenotypes, and biodiversity. Databases may contain empirical data, predicted data, or most commonly both. They may be specific to a particular organism, pathway, or molecule of interest. Alternatively, they can incorporate data compiled from multiple other databases. These databases vary in their format, access, mechanism, and whether they are public or not, whether it is accessible or not. For example, in the other day, we were trying to get the database of FTIR spectra of polymers. And some of them are available open, for example, OpenSpecly. When you want to use the Wiley databases, you need to initially get a bit free, which will not be the the software database in entirety, the spectral database in entirety. You'll have to pay for it the moment you want to license it out. But but luckily, and we are privileged that many of these databases which are used in bioinformatics and network pharmacology are free and found in the public domain without any payments required. Next. Databases in bioinformatics, I would like my colleagues to be mindful. The ones which has been highlighted in red is what I have used in my laboratory. So databases in bioinformatics used in biological sequence analysis, GenBank, UniProt. UniProt we have been using to understand the genes and the corresponding proteins which are acted upon by our molecule of interest. Used in finding protein families and motif funding, Interpro, PFAM, used for next generation sequencing, used in network analysis. For me as a pharmacologist, I'm very much interested to know my molecule or my area of interest, my chemical of interest acts on the genes which actually are related to corresponding which proteins, and that is related to which molecular biochemical pathways, and those pathways are implicated in which cellular activities. Are they involved in biological processes, molecular functions? Are they part of expressing proteins which are part of cellular components like cell membrane, endoplasmic reticulum, mitochondria? All of these are a lot of interest to me. And what we have used, in network analysis in our laboratories, which is mentioned here, and also reactome. Used in design of synthetic genetic circuits, etc., like for example, GenoCAD. Next. Software tools for bioinformatics range from simple command line tools to more complex graphical programs and standalone web services available from various bioinformatic companies or public institutions. My dear colleagues, for example, when I do binding DB studies, to find out similarity indexes of my molecule of interest binding to which genes and understand the corresponding proteins, many times the software doesn't give me the understanding of common genes, target genes, and proteins vis-a-vis their role in a particular disease state. So what I do in my laboratory with my students is I map it manually. So in between interim steps, you may need to map it yourself, okay, using different processes. Next. So the open source bioinformatics softwares are enlisted with their icons: BioJava, BioPython, Embus, Eugene, BioRuby, GenoCAD, and any of us can access these softwares and use it as a tool for our research. Next. This is showing a beautiful diagram. It can be actually a very good pattern in our clothes as well. These are showing what you see in blue and red, either a square or a rectangle or a square or a circle. These are nodes, and these nodes may indicate a drug or a chemical or a protein or a gene. And the lines which you see in blue, right, are the edges. So basically the edges are showing the relationship between the nodes. So the node, as I said, can be any of these factors, and this shows the relationship. The network pharmacology simplifies complex relationships between genes, proteins of interest, pathways, and their role in disease genesis and also progression. Next. So network pharmacology, as I said, we have used. It's used in computational power to systematically catalog the molecular interaction of a drug molecule in a living cell. It is an important tool in understanding the underlying complex relationships between bioactive and the whole body. It also attempts to discover new drug leads and targets and to repurpose existing drug molecules for different therapeutic conditions by allowing an unbiased investigation of potential target spaces. NP analysis not only opens up new therapeutic options, but it also aims to improve the safety and efficacy of existing medications. Need to shift the strategy to more today, as you know, and Ayurveda also was a proponent of it. Our own ancient ethnopharmacological wealth was a proponent of holistic treatment, holistic interventions. And that talks about multiple targets, synergism, and a formulation discovery approach, which may have multiple components. So network pharmacology is a very important tool to understand the complexity of the pathogenesis of a disorder, to understand the potential targets and the ligands which can act on the targets at several levels. Remember, there are certain ligands or a couple of ligands which can act at a cascade of targets within the cell in different biochemical pathways, leading to additive or synergistic outcomes. Next. So applications of NP include drug research, in pharmacology, and also in traditional medicine. The first time what compelled me to look into traditional medicine is I'll give you a sneak peek on some of my research on Ayurvedic composition. I started with, the former vice chairman of UGC and even the former chairman of NAC, Dr. Bhushan Patwardhan's paper, which was, published in around 2017. And it was a wonderful paper on Triphala. And that was the one which sort of, motivated me to take this area up. And we have been using it extensively as a prelude to in vivo studies in my laboratory. Next. So you can see drug research, so many aspects where network pharmacology can be used, discovery of disease-causing genes, studying drug resistance or antibiotic resistance, reducing cost of treatment, understanding the signaling pathway of disease types, etc. Next. Pharmacology, rational design of drugs and groups of interacting proteins, repurposing of drugs, prediction of toxicity. See, when we are talking about two arms of any outcomes, whether preclinical or clinical, there are two major arms which will have its, endpoints. One is the efficacy endpoints, and other is the safety endpoints. So network pharmacology can not only be utilized to understand the efficacy aspects, of a molecule or a ligand to targets and proteins and their involvement in pathways. The same implication can be applied to toxicity, because remember, toxicity also happens because of the drug acting on similar pathways, right? But which is adverse, which is unwanted, right? Next. Traditional medicine, I'm going to tell you how we have used network pharmacology in traditional medicine, that too in a complex Ayurvedic formulation containing 37 ergomineral components. And that was, the thesis was recently submitted by one of my students, and we are in the process of writing the paper, and we will soon be submitting. So traditional medicines, as you see, you'll find many papers from China where they have utilized network pharmacology approach to sort of gain confidence on the rationale behind Chinese traditional system of medicine. The same can be done with Ayurvedic, Indian Ayurvedic, Siddha, Unani, all these forms of medicines which India has been proponent of. Next. Steps in a network pharmacology is very important. This is what we follow. The compound collection. You can use ChemBL, and then you can use the binding DB studies to see your molecule. The similarity score is something which you need to look into, because based on the similarity score, the software will tell you on which target genes it binds. And then you can use that information of target identification to use strings software or UniProt to understand the corresponding proteins which is going to be impacted by your ligand, because it is acting on the target genes. Then comes the next part. You can use KEGG software or DisGenet software to understand all the genes involved in your disorder of interest. One of the disorder which we worked on was nephrotic syndrome. The other was metabolic syndrome. So in nephrotic syndrome, we looked into the entire set of genes, which was more than 1,500. But then we converged and found out our molecule of interest acts on which genes which have been implicated in the disease. So then you narrow down to, let's say, 150 or plus, right? And then you try to understand which are the overlapping and non-overlapping genes if you have a combination of 2 or 3 molecules. And then you do a pathway analysis. The pathway analysis and the gene ontology study, we call it as pathway enrichment analysis, is very important, Because just merely knowing about your ligand acting on a gene and corresponding protein or proteins is not enough. You need to know that these genes are implicated in what cellular process, right? That's the key. And remember, a gene may be implicated in several processes. So to understand the complexity of it, softwares are excellent. These tools give us a simplistic picture of what are the top 5 pathways on which your genes is going to act, the genes on which your ligand is acting, right? And ultimately, you can make a beautiful design of network pharmacology, showing the nodes and the edges using cytosine. Next. Yeah. So as I told you, compound collection is up to you. You can take pharmacophores, you can take phytochemicals, you can take any ligand. You can also use ligand which has been generated in silico by the chemistry lab. I have worked on 2 ligands which were known to be epigenetic modulators in my nephrotic syndrome, in my project. While for my Ayurveda-based project using network pharmacology, I actually identified the top pharmacophores using network pharmacology, which was having a binding DB score of more than 1 to the genes of interest which have been implicated in metabolic syndrome. My dear colleagues, I would like you to know that most of the diseases today, cancer, metabolic syndrome, type 2 diabetes, type 1 diabetes, nephrotic syndrome, other than the pathogenesis which leads to the disorder, there's a lot of role of inflammation, and inflammation in turn also leads to a lot of tissue re-engineering in several of the conditions, one or more. So it's very important for us to understand what are the underlying mechanisms for the disease genesis and also the progression. And for example, low-grade inflammation is a well-known factor in type 2 diabetes, right? We don't take cognizance of it. So what about the effect of the genes which are in inflammation? So when I do nephrotic syndrome or metabolic syndrome, I don't rule out other important allied changes which is happening. For example, dyslipidemia, for example, inflammation. So then you have to strategize as a guide with your students of what you will get in your angle, with respect to the changes. So you should know the genes involved in. We know common genes of TNF-alpha, NF-kappa, beta, interleukin-1 beta, IL-6. So those should be part of your angle when you are doing the analysis. Next. Madhu, can I have the next slide? Yeah, the previous one. I think, the previous one. Yeah. Can I have the previous? I think I've mentioned. Yeah. My dear colleagues, I have used ChemBL for the ChemBL and binding DB studies. ChemBL for the structure, binding DB to understand the gene targets. As I said, you should look into the SCORE, UniProt, and string software to understand the proteins. It is going to be impacted by the genes getting impacted. Reactome and KEGG, that is Kyoto Encyclopedia of Genes and Genomes, I have used for understanding the genes involved in the pathogenesis of that particular disorder. And remember, whatever I've identified in red are available for free, right? These are having public access. Next. So disease information, you can, as I said, I use DisGenet and Reactome. There's another software which some of my students have used, is therapeutic target database. And then as I told you, now I know the genes where my ligand acts. I know the proteins, corresponding protein. I know the genes implicated in that particular disease. Then I do the mapping and converge on the common genes. Next. Pathway analysis is very important, which you can do with PUNE software, or you can do it with Reactome. Here the KEGG pathway is shown. As I told you, once you know the genes, the common genes between the disorder and the ligand action, you need to know these genes are working on which biochemical pathway, and they are working on which functionality of the cell, and that plays a very crucial role in the final analysis, and this is called as pathway enrichment, and gene ontology study. Next. As I told you, pathway analysis and enrichment and gene ontology. I will explain to you when I show my case studies. Next. Yeah. So here you can see this is coming very small in the screen. Then I know my genes, and now I know those genes are implicated in which phenomena of the cell. Because ultimately, when we talk about system pharmacology, it's the body, right? It's the organs composed of tissues, the tissues composed of cells, the cells having several cellular organelles which have their roles. There are several biochemical pathways going on within the cell, extracellular matrix in the blood, which is part of the extracellular matrix, right? And the crosstalk between them. So that's what is the beauty of network and system pharmacology. It is like an orchestra where each percussionist, each singer or musician has an important role to play in the outcome. It can be in health and it can be even in disease. So the gene ontology, if you can just highlight the slide, I'm highlighting because I'm watching in my screen of my phone. The first you can see is molecular processes, molecular function. Then you have cellular processes, and then you have the structure, the cellular, components. So what is the role of the gene of your interest, which has been the common gene, in these phenomena which is happening at the cellular level? And that is very important part of gene ontology and pathway enrichment. So what the softwares do is, you have, let's say, 150 odd common genes ultimately converging, common to your ligand action and common to its role in the disease, right? The software will tell you the top 5 pathways on which these genes are playing a critical role, okay, a key role, and whether those pathways are molecular mechanism pathways, molecular function pathway, cellular biological processes pathway, or they are cellular components. They are part of those proteins, are genes and corresponding proteins are part of the cellular components. Next. Yeah. So what is the importance of gene ontology? Gene ontology provides a system for hierarchical classification of genes or gene products into terms organized in a graph structure. The terms are grouped into three categories: molecular function, biological process, and cellular component, which I just mentioned. And you will get a better idea when I showcase my studies. So geo basically enrichment is a technique for interpreting sets of genes, making use of the gene ontology system of classification, in which genes are assigned to a set of predefined bins depending on their functional characteristics. For example, the gene FASR, which you know is also called as the death receptor, on which TNF alpha and other interleukins will go and bind and will be responsible for apoptosis and necrosis, is categorized in being a receptor involved in apoptosis and localized on the plasma. Right? So whenever we are talking about cell death and necrosis, these are the death ligands would be acting on the FASR receptors. Next. So this is how the classification happens. So the gene ontology analysis tools, there are several. You can see here, Gene Weaver, David, Bingo, G Profiler, Gorilla, nice names to it. Geyser. What we have used is Unrich, okay, and that has really given us good results. And in some cases, we've also used Reactor. Next. Network construction and visualization. Now you have the entire, interplay of genes, corresponding proteins, which are, acted by your ligand. And remember, this is common to your disease condition which you are targeting. You also know these genes, the hierarchy of the genes, and their involvement in cellular processes, which can be biological process, molecular function, it can be cellular component. And then now you have to make your design using Cytoscape, your network. This is where you use a software called as SCES'S. Next. As I told you, the nodes are connected by lines called as edges. You can see a beautiful, very complex structure of a cytoscape on the right hand side, right? Mine will be simpler than this. Cytoscape is a Java-based open source software platform. It's a useful tool for visualizing molecular interaction networks and integrating them with any type of attribute data. In addition to Cytoscape, a number of visualization tools are available. Visual network pharmacology, which is specially designed to visualize the complex relationship amongst diseases, targets, drugs, mainly contain three functional modules: drug-centric, target-centric, and disease-centric visual network pharmacology displays. Next. Our study. One of the study, this study has come out very well, and we are in the process of now coming out with the in vivo data. This was published in Current Research in Physiology. One of my PhD students, when we were kids, he worked on this. What the study entailed initially, this was early part of her work, then she went completely into the in vivo study. A new prospect for the treatment of nephrotic syndrome based on nephropharmacology analysis. Remember, kidney ailments again are difficult to treat. There is no magic bullet. You know, there are multifaceted angles to the pathogenesis, and we concentrated only on nephrotic syndrome because it still remains a bit is an unmet medical need or inadequate therapeutic options available. So our study aimed to identify the potential of two HDAC inhibitors, histone deacetylase inhibitors, which are epigenetic modulators. One was all-trans retinoic acid and others valproic acid, which commonly known to be used in the treatment of epilepsy. In the treatment of, again, see, this is a repurposing of a known drug which is known for epilepsy. The study aimed to determine the combinatorial synergistic potential of the two drugs via network pharmacology approach. We additionally corroborated the result with preclinical setup in a model which is doxorubicin-induced nephrotic syndrome in rats. Next. So what I'm trying to do is that I'm trying to sort of draw a link between what we got in silico and we got in vivo and how it helps in consolidating our claim. So you can see the network pharmacology. In the center, what you see in green is valproic acid and ATRA, VPA and ATRA. And the ones which you can see in the triangles, you know, in reddish color, those are the genes. And what you can see in the blue squares at the very far end of the circle, those are the proteins. So this figure represents the drug network NS specific target. NS is nephrotic syndrome specific target and protein interaction. In the center is drug, outside that is the gene target, and the very outside the rim is the protein targets which these genes are acting on. Okay? The square nodes represent 15 proteins. I told you the edges, the lines which you see in gray, okay, represent interaction between the targets and the proteins. And there also there is angle, etc., which no, I don't want to dwell on now. Next. Yes, so the gene ontology analysis, you can see here how this is what our work in nephrotic syndrome. So from that, have you missed the slide, Madhu? Can you go back? Okay. So here, one thing I want you to notice is there were 25 common targets. 25 common targets between ATRA, VPA, and nephrotic syndrome. Initially, when we saw nephrotic syndrome targets, they were more than 200. So this is what we converged. Now, next, these common targets, which are implicated in nephrotic syndrome and acted upon by both VPA and ATRA, you can see the gene ontology. You see the x-axis, the pathways has been implicated in all the three. The first one, the A, is showing enriched molecular function. These genes, the 25 genes, their role in the pathways associated with molecular function. The 25 genes, their role, and only the top 5 has been shown here. The system will give you 20, 25 more. So you have to select, you know, the top 5 which is having better, the better binding and outcome. The next you can see top 5 enriched biological process pathway. I'm not able to read out in my phone, but I'm sure you would be in your laptop or computer screen. Top 5 enriched cellular components, which is the C figure, right? At the x-axis, you can read out. So you can see it is whether it is a cell membrane component, it is a mitochondrial component or mitochondrial membrane component. So all these things can be depicted in the gene ontology. It shows the importance of these genes in these cellular pathways and phenomena. Next. The same thing is depicted, but we have used two systems here, PUNE and reactome, I told you. So the same diagram which you saw earlier is put in other format, where we use two databases. One was PUNE for gene ontology and the other was reactome, and more or less the results are similar. Okay, and here also you can see the segregation between biological processes, molecular function and cellular function. Next. So 25 overlapping genes you can see which has been highlighted. We can just scroll through it. There are some which are implicated in tissue re-engineering in necrotic syndrome. The kidney gets re-engineered. There is necrosis, right? And next, there is also implication. The role of matrix metalloproteinases, which are playing a role in the breakdown of the connective tissue matrix. Then a role of inflammatory mediators, genes, and corresponding cytokines. So this I'll next come to my in vivo data. So this is just giving you an overview of what we saw and then drawing a link to our in vivo outcomes. Next, Madhu.
Anuradha, you can continue, not a problem.
Yeah, can I have the next slide? Can I have the next slide?
Yeah. And then you please don't hurry. I think you can definitely extend it by 15 minutes.
It was still 11:45
so
now,
don't
my
worry.
time.
You can just go and complete it.
Okay.
Yeah,
fine.
but
Thank
then
you so
you can. That's what I'm saying. You can extend.
Okay. So what you can see here highlighted is where mainly HI is acting or BPA is acting. So what we saw is that transforming growth factor beta, as you know, it plays an important role in tissue re-engineering. See, COPD, you see lung re-engineering. Nephrotic syndrome, you see kidney re-engineering, right? Hypertrophy of the heart, you can see the myocardium, the wall of the heart re-engineering. There are many hepatitis in the long run, NASH, or alcoholic liver cirrhosis. Again, there is a re-engineering of the liver. So there are many disorders where there is a re-engineering of the tissue matrix, right? So transforming growth factors, cytokines, all of these, we caught these genes, in the 25 common genes in our network of parts. Next. This is what an in vivo data is. I'm just giving you a bit of a overview. You can see here the groups: fatal control, necrotic control, HI in different doses, BPA in different doses, and the combination. And we have used a standard, which is a steroid. Nephrotic syndrome steroid is used, for example, prednisone. And there you can see the impact of the combination, which was also depicted in the combinatorial analysis in our network oncology. How much, the commonality of involvement in impact on genes and their impact on proteins which are implicated in the disorder. Okay, so you can see NF-kappa beta, transforming growth factor beta. So you can see here how the combination is showing significantly improved response compared to two very importantly and well-known epigenetic modulators. Next. There also our approach was different. I have not gone into the rationale of using HDAC inhibitors here, which will be part of thesis. MMP2, matrix metalloproteinases 2 and 9, and here you can see that in 2 is a reduction. As you know, collagenases and gelatinases break down the matrix in the tissue and can lead to ease of regeneration. Also implicated in cancer, for example, epithelial mesenchymal transition, which you can see in solid tumors at the advent of the cancer, and plays an important role in the progression of the cancer and the cancer metastasis. So my dear colleagues, this can be applied to many conditions, right? Okay, next. So that was one study. The next study is more complex. As I told you, we were very triggered after 2017 to do something where the components is more complex. So we did a preclinical pharmacological investigation on Chandraprabha Bhatti, which is traditionally known to work in hereditary diabetes, in kidney disorders, as per ancient texts. Our study aimed to integrate NP approach for components of Chandraprabha Bhatti to explore its synergistic potential in multi-component, present with components. Remember, it has both herbs and minerals, but we concentrated only on herbs when we did the network pharmacology to treat multiple complications associated with metabolic syndrome. Metabolic syndrome, as you're aware, is associated with obesity, insulin resistance, glucose intolerance, right? Dyslipidemia, inflammation. See the multiple facets associated with metabolic syndrome. So we thought this is a perfect, platform to do the network analysis. Further, to validate NP results in preclinical setup, we did extensive in vivo study. Next. Yeah. So now you can see it has 57 components, which we narrowed down to 16 because the pharmacophores which we identified, which had a binding DD score of similarity score of 1 was only selected. So these were the components which seem to be highly effective, right? Potent. Andrographis paniculata, Citrus deodara, Tinospora cordifolia, all of these which are part of the Chandraprabha Bhatti composition. Next. It's a very heavy slide. Let me explain. As I told you, 37 carbomineral components. Ultimately, we narrowed down to 16 herbs because of the binding DB studies, ChemBL binding DB studies, unit brought the scores, and then in that also we identified the pharmacophores of interest. It's a huge set of data. So when we took the print and we were studying it, it's like 500 pages of data, right? But simplified by the software, right? And what was the disease genes which we consider? So we narrowed down. As you can see, we narrowed down the genes which were associated with 16 of the components and their pharmacophores. The disorders, we had 3 angles to it, like in nephrotic syndrome, only 1. Here we saw dyslipidemia, we saw inflammation, and we saw metabolic syndrome genes, right? So this is, and we tried to get the common genes. Next slide. We did the mapping and did the common genes. Here you can see the figure. This is a cytoscape-driven network pharmacology figure. It represents drug, target, and protein. The one which you see in pink, right? And the yellow are the components. Center pink below is Chandraprabha Bhatti. The components of Chandraprabha Bhatti is in the yellow nodes, okay? And their link to the genes are in pink and orange, and the pink related genes and the corresponding proteins are in green. The orange related genes and the corresponding proteins are in blue, right? So this is what, and you can see there are 33 overlapping genes. What does that mean? 33 genes, and this is for metabolic syndrome. Separately, we did for inflammation and for dyslipidemia. 33 genes common to the components which are mentioned here, on 11 components, their pharmacophore, their action, and implicated in metabolic syndrome. Okay, next slide. This is showing the drug target is gene and protein interaction network specifically for the 33 overlapping and common target genes involved in Chandraprabha Bhatti for metabolic syndrome. Triangle nodes in pink represent the 33 putative targets specific for metabolic syndrome, and you can see all these herbs mentioned, okay, which you can see in these yellow nodes. Square nodes in yellow, okay, color represent the 66 interacting proteins, which is at the outer rim of the circle. So the proteins, okay. The genes are the ones in pink triangles. The edges represent the interaction between the CPV and its constituents, targets, and the interacting proteins. Next. Now, same like we did for the study on nephrotic syndrome, we also did the study on for gene ontology and enrichment of the pathways. We did it for Chandraprabha as well. And here you can see the figure representing the gene ontology, the top 5 A, B, C figures. A represents enriched molecular function, the genes which are participating in molecular function, top 5 in A. In B, the genes involved in biological processes, top 5 in B figure. And in the C figure, the genes encoding for proteins which are part of cellular components. And this is specifically for the Chandraprabhavati pharmacophores, right? So it gives a lot of picture on the importance of these. So it also, my dear colleagues, helps us justify and rationalize the ancient Ayurvedic compositions, which were made by our Vaidyas those days, without understanding of molecular pathways, without understanding of targets, but they knew about the basic tenets of Ayurveda, right? And they knew it worked. And today, with using network pharmacology, using molecular biology approaches, we can consolidate and take pride in the Ayurvedic rich texts and compositions in India. Next. The same pathway analysis, which has been the gene ontology pathway, has been depicted in a different figurative format. And the same thing, as here also you can see, which are the genes which are implicated in these of the cellular phenomena. Next, this is just a way of depiction. Like I showed you for nephrotic syndrome, here also it's a huge set of data, right? But I would like to tell you how these databases, you can see protein kinase C binds on activating lipases, which will be implicated in fat mobilization, especially the external, the internal pathway, chymomicrons getting absorbed, going to the liver, getting processes to form the lipoprotein particle. There's so many interplay, right? So you can just scroll, Madhur, we can, my colleagues can just see, and I'm just giving you a very brief peek into it. As I told you, the data is enormous. Next. But it is very easy to simplify the data when you are presenting in your publications. Next. The overlapping genes, PPAR alpha, we all know. HMG CoA reductase, they are the drugs which are used. So, PPAR alpha, their HMG CoA, these are drugs these are targets which are utilized by anti-hyperlipidemic drugs, right? And see, the components of Chandraprabhavati have impact on these. Next, and that will impact fat mobilization. It also has impact on inflammatory markers. Fatty acid binding protein, again, very important, FAP4, right? We have worked. So subsequently, when we did the in vivo study, we got results which concurred with these findings of gene targeting and the protein implication. Next. This is the Chandraprabhavati network construct, right? And this is for inflammation. The earlier one was for metabolic syndrome. I am not showing you dyslipidemia because of paucity of time. So metabolic for inflammation, you see the number of pharmacophores and corresponding herbs has reduced. There it was more than 11 or 12. Here it is less than 10, right? Which are implicated in common genes, 14 overlapping genes, 7 non-overlapping, which have been implicated in inflammation, right? Okay, next. So here again, you can see that the green, Madhu, 1 second, just go back. The one which is the lower circle, The pink is Chandraprabhavati. The one which is in orange notes are the components, the herbal components of Chandra, like terminalia, cellula, and other components, whatever the components is. And those components acting on which genes? The common ones are in the common genes, overlapping genes are in green. The non-overlapping genes are in violet. The corresponding proteins are in blue triangles and in yellow nodes. Okay, these are all nodes. Yeah. And you can see the gray edges which shows the name. Next. This is highlighting only the the common targets, the overlapping targets. In the center is Chandraprabhavati in red. The yellow mustard colored nodes are components of Chandraprabhavati. The corresponding genes, which their targets, which is in green triangles, and light green, the very rim of the network is the proteins. Next. These are, you can see here, PPAR alpha, next, which are implicated in inflammation as well. Then, next, glucocorticoid receptor. There are many we saw which we identified. And then during our in vivo studies, we did several endpoints which were related to inflammation. We also did insulin resistance, HOMA-IR, so fasting insulin, fasting glucose. All of these were done by us. I have not shown you the results. My dear colleagues, we did a lot of work on mitochondrial function because mitochondria, as you know, it maintains cellular energy homeostasis. There's an important role in the kinetics, energy kinetics of the cell. And we did a lot of study on PGC1 alpha, TPAM. We did RT-PCR subsequently. So what happened is, when we really estimated the gene expression studies in vivo, and then we could sort of draw a link to our network pharmacology outcomes. Next. Madhu, can I have the next slide? Yeah. Now, my dear colleagues, thanks, Madhu. This is what, I'm giving you a peek of what we have done in vivo. So it's not just network pharmacology, you'll find it very difficult to publish your work. So you have to follow it up with in vitro work on mammalian cell lines or some cell lines, whichever, cancer cell lines, etc., or you do in vivo. So here you can see the expression changes, PGC1 alpha, master regulator and orchestrator of mitochondrial biogenesis. Mitochondria is like, is the only in the evolution of mammals. Mitochondria is said to have come with from eukaryotic microorganisms which needed oxygen to survive, right? There are two species, and there were some other unicellular organisms which had something like chloroplasts, which led to formation of the flora in evolution. While in evolution of mankind and other mammals, these the mitochondria came from unicellular organisms. Life form emerged from the oceans, right? And as we know that other than our nuclear genome, mitochondria has 13 genes, and they are important. There are no other cell organelles other than nucleus which has their own genes. Mitochondria has its own 13 genes, and they are responsible for expressing for proteins and enzymes, which are responsible for the electron transfer, right? They are an important role in energy, cellular kinetics and homeostasis. And if there's a disruption of mitochondrial function, there's also role of inflammation downstream, inflammasomes. It's a well-known factor now, right? So we have done TPAM. Right? TPAM is transcription factor A of mitochondria, and here you can see the role of Chandraprabhavati, and and the standards which we have used is 2 because of the complexity of metabolic syndrome. One we use fenofibrate, the other we use metformin. Fenofibrate known to be a drug for dyslipidemia, and metformin is a drug used for diabetes. So that we could look into both the angles and which Chandraprabhavati for both these drugs which are implicated in dyslipidemia, obesity, and intolerance to glucose. Next. Oral glucose tolerance test, serum insulin, HOMA-IR, to show insulin secretion, etc. All of these, and you can see what we got from network pharmacology approach. We could concur with those results in our in vivo outcomes, efficacy outcomes. And we also did a bit of RT-PCR and those kind of studies, and it helped us augment our claim. Serum cholesterol try. So we did the entire lipid profile. We did the carbohydrate metabolism with OGTT, with fasting insulin, linked fasting insulin to fasting zero time point glucose, found out HOMA-IR, did inflammatory markers, TNF-alpha, interleukin-6, 1-beta, etc. So just trying to show to you that how network pharmacology helped us understand the impact of the multiple component of Chandraprabha. Next. So in conclusion, network pharmacology analysis presents an immense scope for exploring traditional knowledge to find solutions for current problems challenging the drug discovery industry. Network pharmacology can play a role in drug discovery, repurposing, and rational formulation discovery. Many of the bioactive target combinations have been experimentally studied. The data synthesis using NP provides information regarding the mode of action of traditional medicinal formulation based on their constituent bioactives. This is a kind of reverse pharmacology approach to deduce the molecular mechanism of action of formulations using modern integrated technologies. NP aids the logical analysis of the system that can be utilized to understand the knowledge as well as to invent novel solutions for current pharmacological problems. For Chandraprabha work, we remembered the words of Dr. Ashok Reddy when he kept on saying reverse pharmacology when we came to Ayurveda, traditional systems of knowledge, in medicine in India. Next. Next. I have pictures of my lab and I want to show it to you all. Some of the references, this is my students. You can see them, in my lab, my MPharm and PhD students. Rini is one who is in the maroon, this on the left side, this in standing, on the other side corner. The one who did Chandraprabhavati work, Prashant, is on the other panel behind in a navy blue kind of shirt. They are the ones who worked on it. Next. Yeah, this is once we had gone to BARC to present one of our papers on mitochondrial dysfunction. I've been working on mitochondria since almost more than 10 years. See, beautiful, my lovely students, my lab staff, my colleagues. Yeah. Thank you very much.
Thank you, ma'am. The session was very excellent and full of informative. Now the session is open for question.
I would like to thank Madhur for changing the slides. I know it must have been this, but you can't help it. Just suddenly it happened, and I thought better find we pharmacy people and our leaders like Patravale madam. You have to be cool and see to it that you give your outcomes.
Yes. No, but I mean, see, in real life, everything is out of syllabus, no? So this was out of syllabus. Yeah.
Can you repeat what you said?
Yeah, you managed very well. I mean, excellent presentation.
I think there is an ego from Madhur's side.
I mean, I have put one question there. I mean, I think ma'am will take that up. I mean, she can take up the question.
Josa ma'am, there is a question you can ask.
Hello. Yeah, Anuradha. So I
Yes.
said it was an excellent session.
Thank you.
And I have put a question. I have put a question in the chat box.
Madhur, it is echoing. I will ask
Um,
the question on your behalf.
Madam will take it up.
Yeah,
Ma'am,
Jyotsna ma'am.
are you there to take
Yes,
the
I'm there. Don't
question?
worry.
Okay.
So her question is, did you get a training on these softwares or you learned
Jyotsna
all
ma'am?
by yourself?
Yeah.
We learnt it ourselves, ma'am. Yeah, self-help is the best help. Yeah.
One more question is there, ma'am.
So I
can-
think probably
Yes.
there's some problem for
Hello.
Yes, Jyotsna madam.
Ma'am, one more question is there. Can you please share the databases and softwares used for gene ontology analysis? And is there any
No, ma'am.
demo?
So I think I'll only ask that question.
You can get FundRich online. You just have to Google for it.
And it's
You can download. Yeah. All the softwares which I highlighted, they are available for free. We have not licensed it.
Okay, ma'am.
Hello, ma'am. Hello.
Yes, Manisha.
Ma'am, in slide number 51, you showed the comparison of in vivo. So my question is that in network pharmacology, we are mainly targeting the ligand or something. But in vivo, we are going for the dose dependent. Then how we can compare?
We are talking about here the molecular pathway, madam. So the
Okay.
comparison is on molecular pathway. What I am claiming, so for example, in a complex disorder like metabolic syndrome, where there are many facets to the disease, there are implication of several pathways. It can be lipid metabolism pathway, right? It can be carbohydrate metabolism pathway. It can be inflammatory pathways, right? There are several pathways involved. So when you do network pharmacology, you are able to get in silico the targets, right? For the genes which are implicated in the disorder and the genes on which your pharmacophores or bioactives are acting. And then when you have got that and you've narrowed down to the common genes, and you've also done the pathway enrichment and gene ontology, you know the involvement of those genes in which pathways, or cellular component. Then when you do in vivo study, it becomes more it bolsters your claim. Your in vivo data will be bolstered if you get similar outcomes in your network pharmacology. And remember, that's more important because we are looking about holistic intervention.
Okay.
And that's what today's therapeutic strategy is all about: customization, personalization, holistic treatment, right? So this helps.
Okay. Thank you, ma'am.
Thank you.
Hello.
Dr. Anuradha, like you just mentioned that our ancient Vaidyas, I mean, the way they have made these formulations, though they didn't have any knowledge of molecular biology and all, but they managed to get the right formulations. So how do you think they must have, as in, I mean, it's just my, my curiosity and just my, just a question, like how they must have done this in that era.
I think it must have been run by practice. So it must have been,
Okay. Yeah.
over, let's say, 5,000 years of 5,000 years of dissemination of the information, because those days herbs, many of them were not cultivated. They used to carry it and forage it from the forests. So they knew something works. Some were from tribes, some were from the settlements,
right,
of those humans, which they knew that this worked. It must have started with simpler this, because those days lifestyle was different, wound healing, a snake bite, those kind of.
Right.
But over time, for example, metabolic syndrome, the concept itself did not exist during Susruta's time. right? We are suffering
Yeah.
because of our
Yeah,
sedentary lifestyle and,
exactly,
and our food, the kind of processed food which we're eating. Processed food was minimal. There was no
Correct,
processed
correct.
food those days. So
Yeah,
it's
right,
tight of getting an analogy of what was, it was proposed more for kidney related ailments, Chandra Prabhavati, right? And a bit for Madhumaya
Okay,
also.
okay,
And that's what drove us to, because there were
okay.
some pharmacophores which have been reported, bioactives reported to be effective in diabetes, type 2, etc. There were some studies, right? So that's how.
Right, right, right. Yeah, but it's phenomenal, right? I mean, if we try to see those things in retrospection, it fits perfectly well.
Yes, it does. It does. And I'm hoping once my publication comes through, I'll share with you all.
Yes, please. Yes, please.
Of nephrotic syndrome, you can get it online. For Chandra Prabhavati, whenever it gets published, I will share with you.
Sure, sure. We will be looking forward to having it. Yeah.
Thank you. Thank you very much.
Yeah.
Is that all? Is there any other question? Thank you.
Well,
Thank you,
I think
everybody.
No, no. Yes, Josa, you can take it forward now.
Yes, ma'am. Thank you, ma'am, for informative session. Ma'am, you have given the information regarding the bioinformatics, drug discovery, molecular docking, various softwares and databases in bioinformatics, protein target drug delivery, disease information, pathway analysis and enrichment, geno-ontology analysis and enrichment, geoanalysis tools. Definitely all the participants would benefited with this and will transfer this knowledge to their budding students. Thank you, ma'am. I would like to thank from the bottom of my heart to spare your valuable times with us. Thank you, ma'am.
Thank you very much. Thank you for having me. Thanks to APTI, Maharashtra State and Women's Forum. Thanks to Dr. Vandana Patravale ma'am, and thanks to Anaga Joshi ma'am for inviting me. Thanks to all my colleagues from Indira College of Pharmacy and all colleagues who have attended my session. Thank you very much.
Excellent session, ma'am. This has actually opened a new way of standardizing all our traditional medicines in a different way. Yeah.
Yes, certainly.
Lot of work.
And the Chinese have been doing it relentlessly. Why
Yeah.
not us?
Why not us? Absolutely. It's excellent. Very good.
Thank you.
Looks like I missed a very interesting session, but I'm looking forward to YouTube session to get a feel of it.
I was referring to you, ma'am. Thanks for your leadership and thank you very much for this entire concept of Women's Forum of APTI. I think this has opened new vistas for all of us.
Absolutely.
Yes.
Thank you.
Thank you. Thank you very much.
You all. Yes. Josa, you will like to sum up?
Yes, ma'am.
Yeah, please.
Okay, thank you, ma'am. During this 3-day webinar series, we had eminent speakers from different colleges. On first day of a webinar series, first session was taken by Dr. Fariza ma'am, Principal Consultant Pharma AGC. Ma'am has given the session on artificial intelligence and machines. Ma'am has given valuable insight regarding the importance of AI and machine learning in research, how we can speed up our research by using this artificial intelligence. Second session was taken by Dr. Aarti Prabhu ma'am, Associate Professor, Dr. Bhanuben, Nanavati College of Pharmacy, Mumbai. Ma'am has given session on new drug discovery by using the artificial intelligence. Ma'am has given the information regarding the structure-based drug discovery and ligand-based drug discovery. Second day of webinar series was, first session was conducted by Dr. Ruxana ma'am, Professor, Associate Dean, School of Pharmacy, GH Raisoni University, Amravati. Ma'am has given information regarding the innovative pedagogies. She has given the tips how we can increase student learning interest by implementing these innovative pedagogies. Second session was conducted by Dr. Sunila Dhaneshwar ma'am, Professor and Director, Amity Institute of Pharmacy, Mumbai. She has given the session on scientific writing and plagiarism. She has given information regarding how we make our scientific writing clearly and concisely. And today we have Dr. Indupal Kaur ma'am. Ma'am has given the information regarding translational research, how we can make our collaborative work, our research work, how we can make our good laboratory practices by implementing the various research methods and methodologies. Dr. Anuradha Majumdar ma'am has given the informations regarding the drug discovery by using the various databases and software. So I must my heartfelt gratitude to all the eminent speakers for their valuable guidance and inspirations. All sessions were full of knowledge, and we got the knowledge how we write a scientific work, plagiarism, how we make teaching learning process more interesting and interactive, how we can enhance the research process by using the AI and various softwares. Definitely all the teaching fraternity would be benefited by this webinar and will percolate this knowledge to the students for their betterment, and will implement this in teaching and improve the teaching learning process. My sincere thanks to Dr. Rakesh Sumani sir, President APDI Maharashtra State. My sincere thanks to Dr. Vandana Patravade ma'am, Dr. Annagha Joshi ma'am. I would like to thank to all the participants for their overwhelming response. Thank you. It's my kind request to all participants to fill the feedback form. The feedback link is given in chat box. After submission of feedback, you will get the certificate. Thank you.
And I will also, being a convener of this program, I will also like to express my gratitude to all our organizing committee members, Manisha, Dr. Manisha, Josna, Lovina, Neha, and of course, Dr. Supriya Sidhaya and her team for coordinating with for the certifications, and of course to the team of Indira College of Pharmacy who have put in great efforts, like Madhur and Suvarna and Dr. Digambar Ambekar also. And yes, like we will continue arranging such sessions if all the participants are really interested in doing so. And this was the first program organized by APTI Women's Forum Maharashtra State. And thanks to Dr. Rakesh Somani, who has actually inspired us to go ahead to do this program. So thanks a lot, dear team, and your respective colleges for making this happen. Okay, so like you can leave this. So we are concluding this seminar now. If you have filled up with the link, then you can leave the. I'm keeping it open for some time till everybody fills the link properly, and then we will be out of this Zoom link. Okay, so please fill up the link to get your certificates.
Thank you, ma'am. With the permission of convener of the webinar series, I declare that the program is over.
Yes.
Heartiest congratulations, madam, taking the lead. It was wonderful.
Thank you, ma'am. All grateful to you because, as you said, that you have initiated and given a different face to this APTI Women Forum, being a national convener, and we all would love to continue working under you for the same. So thank you so much for your presence. Is so inspiring that we always think, "अरे मैंने इतने किया है." आप वहाँ होके भी हम लोग को inspire करने के लिए you are present here, and that's a lot motivating to us, ma'am. Thanks a lot.
Yeah, let's work together. No one is under anyone. Okay, please.
Yeah, no, but this leadership we have to acknowledge. Otherwise, we become lethargic sometimes. Ki nahi karte hai.
Yeah. So let this webinar series continue, I think, and now we have to see that how we can take it to the national level. Though you did get good participation, I feel that we can reach out to more.
More, definitely. Yes.
Thank
I think the timing should be when all the exams will be over, the teachers will be little free to attend. And because now when I called up many people, because of the exams going on, though they wish they couldn't attend. So our timing probably was little bit wrong, you can say. But
I think this will continue. Some of the other activities always are there. So we should continue irrespective of, some people are able, majority of the people, if they are able to join, that serves our purpose.
Yes.
And once they know about the programs conducted, I'm sure that many more would wish to join. So
Correct.
let that the flow. It's already begun. Let that continue.
Ma'am, my request is like we have been arranging the seminars which are actually theoretical. So if we can come together and arrange seminars at prominent places, but with some hands-on experience.
Sure, absolutely. That is what is my plan too. In fact, we have discussed it at the center level, and they are very keen. So that should be done wherever there is expertise. It should be done. One has been pending.